Okay, hello everyone. Welcome to our last day of Learning Labs with ANH Academy Week 2021. My name is Elena Martinez, and I'll be the technical manager for this session titled Strengthening Causal Inference from Randomized Controlled Trials of Complex Interventions. Thank you so much for taking part in ANH 2022. We look forward to a robust and interactive session today. As we get started, I want to remind you that you can find more information about the ANH Academy Conference, as well as materials and our program on the website. And before I pass things over to our facilitators, I want to make a few technical announcements. First, this session will be recorded and it will be posted on the ANH website following the conference. You've all been muted as you come into the room, but feel free to introduce yourselves using the chat function, and we'll have plenty of opportunities for interaction during this session. We encourage you to turn on your camera if possible, though you can participate either way. And finally, if you have any technical problems, we encourage you to check your audio settings and your internet connection, and you can also message us in the chat box. We have a variety of support people here in the session, and we can help you troubleshoot any issues that come up. So with that, I would like to pass it over to Shelley Sundberg to get the session going.
Thank you, Elena. Good morning, good afternoon, good evening to everybody. Thank you very much for joining us. It's great to be back at the ANH Academy 2022 edition. We would like to speak to you today about conducting randomized controlled trials for complex interventions in agriculture, nutrition, and health. And I'm going to be your moderator today. I'm Shelley Sundberg at the Bill and Melinda Gates Foundation. I co-lead our work on nutritious food systems and women's empowerment in agriculture. Next slide, please. And today, presenting this session will be Jeff Leroy, who's a senior research fellow at the International Food Policy Research Institute. Ed Frangillo, who's a professor in the School of Public Health at the University of South Carolina. Bezawit Kase, who's not going to be presenting, but was a co-author of this paper, an important contributor to this work. She's a PhD candidate in epidemiology at the University of South Carolina, and I'll be facilitating the session. Next slide. The objectives for this learning lab, our overall objective is to address key questions, hopefully that you have from your own experience, about the design, implementation, analysis, and interpretation of randomized controlled trials for complex interventions. And we'll be, a little bit later in the session, we'll be defining what we mean by complex interventions. And more specifically, the objectives are to understand what are the three basic design principles RCT studies should adhere to. And then we'll define primary and secondary outcomes and how causal inference differs between both. Then we'll talk about how to manage the multiplicity of outcomes required in trials of complex interventions while trying to minimize false positive and false negative findings. And then finally, why multiplicity should be managed through careful planning and interpretation rather than addressing it post hoc through doing adjustment in the analysis stage. So those are the objectives of the session, and we hope it will be interactive and you'll have an opportunity to ask your questions and participate in a breakout session as well. Next slide. I'll go through now the plan for the Learning Lab. The first part will be to understand your questions and experience in designing and implementing and interpreting randomized controlled trials. So we'll elicit those questions and experiences from you. Next is we'll talk about the use of randomized controlled trials for impact evaluation. Next, we'll look at what does it mean to have RCTs for complex interventions. What do we mean by complex interventions with multiple pathways? So we'll introduce those and do some group work around those. And then the final part of the first part of the session is to talk about the accuracy of inference and the inappropriateness of some typical recommendations, which are more geared towards simple interventions. Then we'll take a brief break and come back and talk about the three basic principles for RCTs of complex interventions. Then a typology of outcomes, and again, talking about causal inference. Then we'll talk about multiplicity, what it is, how to address it. And then recommendations overall. What are the 7 recommended steps to implement randomized controlled trials of high scientific integrity? And then finally, we'll have an opportunity for you to ask questions and hopefully be able to provide answers. And I just wanted to say, as the session is going on for all the sections of it, please feel free to put your questions in the chat. You don't have to wait till the very end, and I will monitor those, and hopefully we'll be able to get to most or all of them during the question and answer portion of the lab. And next one. Thank you. So first, let's start with trying to understand what your experience has been in designing and implementing and interpreting randomized controlled trials. We're going to do a poll. I'm going to turn it over to Elena, who's going to run the poll. But first question is, what is your experience with RCTs of complex interventions? Do you have no experience? Are you brand new to them? Have you read papers about RCTs but not conducted them yourselves? Or have you conducted RCTs but not as the PI? And then maybe some of you have been the PI on an RCT. So we'd love to see what your experience has been. And then the second question is, what are your top 3 questions or challenges you faced in the design, implementation, analysis, or interpretation of RCTs of complex interventions? We've had similar sessions like this in the past few years at ANH Academy. And I know a lot of you have had a large number of questions, really interesting, provocative questions that you've encountered in your own experience. So we'd like to hear those. So Elena, over to you to conduct the poll.
Great. So we're going to do the poll through Poll Everywhere. I've put the link to the poll in the chat, so you can click on that link. And I'm going to share my screen so that we can see the poll question together. So we'll start with the first question. What is your experience with RCTs of complex interventions? I've read RCT papers but not conducted them. And many of you have conducted RCTs but are not the PI. Okay, now we'll go on to our next question. What are your top 3 questions or challenges related to the design, implementation, data analysis, or interpretation of RCTs of complex interventions? Okay, it looks like people have questions about calculations, about data management, about the ethics of doing RCTs. What else? About translation, power, and analysis. Okay, I'm going to stop sharing. It looks like there's a lot of different questions. So back to you, Shelley.
Thanks, Elena. And I just wanted to say that the poll was great, so everybody could see sort of the issues that people are interested in addressing. But if you would like a specific question answered during the session, again, please feel free to put it in the chat at any time, and we will try to get to it. I'm going to just, one thing I wanted to emphasize, that there's a paper that's come out of this process that Jeff's going to show toward the end of the session. But I want to emphasize that some of you maybe have attended previous sessions where we discussed randomized controlled trials, including the last in-person ANH Academy Week in Hyderabad, about 3-4 years ago. And since then, there's been a very consultative process that has included consultation with people coming from multiple disciplines. So it's been a fairly long journey, and a paper is now published offering this guidance. And so that's what we're going to present today, and we look forward to a really interesting discussion with you. So over to Jeff. Thank you very much.
Thank you, Shelley. Welcome, everybody. It was good to see in the word cloud that we will be covering some of the issues that you identified in the presentation, but for other issues, as Shelley said, feel free to put them in the chat, and we'll try our best to answer them at the end of the session. So for those who have little experience with RCTs, this is a very quick high-level overview of what an RCT is about. So RCT stands for a randomized controlled trial, and we use them for impact evaluations. So what is the impact of an intervention is a basic question we're trying to answer using this tool RCT. And impact means, if you want to use an equation, it's the outcome you have when you have the intervention, and there is a minus missing there, minus the outcome without the intervention. If you don't like equations and you just want a visual representation, it's shown in this little table. So imagine you have an agriculture intervention with cows, and I believe it's a chicken there, and you want to assess what the impact is on child dietary intake. You would have a group that is exposed to the intervention and you assess dietary intake, and you have a group that is not exposed to the intervention and you measure the intake as well. You take the difference of the two, and that would be the impact of your intervention. The problem is that you can never observe what happens to a person when he or she receives the intervention and what happens to that person, the same person, when she or he does not receive the intervention. You cannot observe that in the same individual because you have to choose whether the person will or will not receive the intervention. And that's the central problem in impact evaluation, and that's why we need a counterfactual, which is basically what would have happened in the absence of the intervention. Now, RCTs provide an elegant way to create that counterfactual. We're going back to the example of the agriculture intervention. We have a number of children who we want to include in the study. We toss a coin to basically decide which child will receive the intervention and which child will be in the control group, not receiving the intervention. And as you can see here in this simple example, when you look at the colors, which could represent characteristics of people, maybe their poverty level, the parents' knowledge about nutrition, we have a nice balance between the two groups. You have a bit of red and green and gray blue on both sides or in both groups. And that's exactly what randomization is used for. So randomized controlled trials are considered the gold standard for impact evaluation. And why is that? Because if they're done well, you can assume that the intervention and control arms are comparable. That means that the only difference that is left between the arms is the actual intervention. And that means that the control arm now provides a valid counterfactual for the arm exposed to the intervention. So that means in conclusions that any differences we see in this particular example, the diet of the child, should be due to the intervention. We know that there has been quite a bit of debate about RCTs, about issues related to ethics, about the adequacy of using this tool for impact evaluation. And many of these discussions are useful, but in this session, we will not offer any guidance on when RCTs are appropriate. So the focus of today's session is on, once you've decided to use RCTs, how to best use them and to extract solid evidence out of them. Back to you, Ed.
So this work was motivated by the realization of people doing evaluations of the impact of complex interventions, that although there's wonderful guidance out there from the Consort, for example, and other places, that not all of that guidance was appropriate or provided the kind of help that investigators needed to make decisions about complex interventions. So what's the difference between them? So a randomized controlled trial of a non-complex intervention aims to assess the impact of a well-defined, typically single intervention, which might be an antibiotic treatment, micronutrient supplement, anything like that, on a narrowly defined outcome, which might be the number of days ill, micronutrient status, something like that. So trials, for example, that are assessing whether a drug is efficacious is an example of a non-complex intervention. An RCT on the right of a complex intervention assesses the impact, in contrast, of interventions that are composed of two or more, often many, intervention components, ranging from behavioral change communication, gender sensitization, micronutrient supplements, cash transfers, a wide range of possible components coming from multiple disciplines or sectors on a wide range of outcomes across different domains. Next. So for non-complex interventions, typically on the left, bottom left, that they're based on, most often based on a solid understanding of the biological mechanism, which that understanding is required to justify doing the trial to begin with. But for complex interventions, the exact biological and behavioral mechanisms are often not known. And so we have theory about how the intervention will work, but we don't know exactly how the intervention will work. And part of what we're trying to learn in doing the trial is how does the intervention work to create impact, as well as does it create impact. And so there's an interest in estimating the effects on a range of outcomes that are linked to the objectives of the intervention, the end objectives of the intervention, but also intermediate outcomes along the theorized impact paths that are intended to generate those end outcomes. And so because of this, limiting the number of trial outcomes to one or a few runs the risk of missing important results. And so that's one of the essential sort of challenges that we're going to be tackling today. Next. So here's an example. This is a trial that we recently completed in Western Kenya. It's a complex intervention. The inputs for the intervention are on the bottom left there, which is a microfinance loan providing a foot-powered pump to generate water for irrigation and other uses. Other inputs like seeds, and then a training component. So it's an agricultural livelihood intervention. And the theory of change in this study was that those would improve food security and household income and wealth, that those in turn would lead to better nutrition, better adherence to antiretroviral therapy, because this is an HIV, a population where HIV is prevalent, improved mental health, and enhanced empowerment. And those in turn would affect the sort of clinical end outcomes of decreasing morbidity and mortality related to HIV. So you can see that there's, even in this simple diagram, which is hiding much of the complexity, that there's a lot of different boxes and therefore a lot of intermediate as well as end outcomes. So next slide. Oh, and so please use your phone to take a picture of this diagram, because you're going to need it for the group work that's going to follow shortly. Okay. So, all right, moving forward. Then for the group work, we're asking in the breakout sessions, three questions. Which outcomes would you want to assess? So based on that theory of change that I just showed in the figure, what outcomes do you think you would want to assess? What are the important outcomes? There's no need to think about the tool to measure those outcomes. Just name what the outcomes are. So for example, food security is an outcome. It's a construct, if you like. And the household food security access scale might be a tool to measure that. We're interested in the outcomes. The second question is, which of these outcomes should be the primary ones? Which are the ones that you would want to declare as the primary ones? And then the third question is, say something about how difficult was this exercise to determine what are the outcomes and then which ones of those should be primary. So that's what we would like you to work on. And if you could take a photograph of this as well, then we can stick the questions in the chat also, but take a photo of this, and then we'll be moving in just a moment to the breakout rooms.
Okay, are you ready to open the breakout rooms now?
Yes, please go ahead.
Oh, can you just read through this, Ed? So people know, well, I can, since I'm speaking, I can quickly do it. So you will be taken automatically to your breakout room. You don't have to do anything. The breakout session will last about 10 minutes, and you will also be brought back automatically. And it's good at the very start to identify just one person who will report back on what we asked you to do. And the reporting back will be very brief. Thank you. Elena, over to you.
Okay, great.
Hello, please, can you please go back to the diagram? I want to take the picture.
Sure.
Yes. So if you didn't have a chance, Ed is showing the diagram again right now. I've also put some quick screenshots of this theory of change diagram and of the questions for the breakout rooms in the chat, so you can click and download those there. In just a moment, I'll open the breakout rooms. You've each been assigned and you'll get a notification on your screen to go join a breakout room, or you can click the breakout icon at the bottom of your screen and join the room to which you've been assigned. And if there's anyone in the main room who's having trouble figuring out how to join a breakout room, just unmute or put a question in the chat and we'll try to help you.
Elena.
Yes.
Just
Hi,
Hi,
Sunita.
Sunita.
hi. I'm just hopping here and there.
Would you like to join a breakout room, Sunita?
Would I? Yeah, sure. Put me in something.
Okay, I'm going to
Hi,
send you
Shelly.
over to
Good
one.
to see you. Hi, Ed.
Hi, Sunita. Nice to see you.
Nice to see you too. Okay, I'm joining a breakout group.
Enjoy.
Elena, did you put a timer on the breakout rooms, or should I put my timer on my phone for 10 minutes? So should I just tell you?
Yes, that's good. There's no timer, but we can close them in 10 minutes.
Okay, sounds good.
Jeff,
If I join, go ahead, Ed.
no, I was going to say, maybe with 2 minutes to go, you can broadcast a message to, or maybe 3 minutes to go. You can
Okay, I'll
click
do
on
that.
broadcast message to all and tell them 3 minutes.
Okay. Will do.
And if I join one of the rooms, I can come back, right?
Yeah, as co-host, you have the ability to move in and out of any of the rooms.
I'll take a quick peek to see if people are not too confused about what we asked.
Do you want us to do that too, Jeff?
Sure, I'll start in room 1 and maybe Ed room 2 if he's up to it, and Shelley room 3.
Sure.
Just noticed that there are a few participants here, I think. Paula and Nilufer. Would you like to go to a breakout room? Should we just assign them?
If you're having trouble figuring out how to join your breakout room, just let us know.
You probably will have something popping up and you need to click join.
Sometimes you also need to just go to the breakout room icon at the bottom of your screen, but I'll try to send each of you another notification. Anna, sometimes if you just move them to a different room, it will send another notification.
It's quite busy.
Yeah.
It's good.
Mm-hmm. Hi, Jane. I see that you've just joined the call. We're in breakout rooms right now, so I'm going to go ahead and assign you to a room and they can help you catch up on the activity.
Okay, thank you.
Great. See. You should have just seen an invitation pop up on your screen to invite you to join room 2. Oh, that worked. Okay.
Can the participants see the chat if we post things in the chat?
We can broadcast a message to them.
Because yeah, Joe and Steph shared this Google Docs with the screenshots. It could be useful.
Oh, I see. They're asking. I can let let me see if I can give I think I can give Joe, well, through a breakout room, I can't. The way our accounts are set up through Tufts, we can't allow all participants to share screens, but the co-host should be able to.
Oh yeah, the co-hosts are in the rooms, right? So they
Okay.
went to see if they were okay.
Okay. Did you share that?
I only pop in the chat, but I'm not sure
I'm
if
gonna
we can
broadcast
do.
it to the breakout rooms. I think you shared it with the waiting room.
Jeff is back.
Hi, Jeff. Just sent a message for 3 minutes left.
Wow, it goes by fast.
You're muted, Jeff.
Oh, I said great.
Okay, that's great.
I'll go to one more room, see how it's going.
Okay.
Are you putting a timer in the breakout rooms, or are you just closing them when it's time?
I'm just going to close them when it's time.
Okay.
I find people usually change their mind about how long they want the breakout rooms open, so I don't use the timer.
Good point. It's true.
They're all doing well.
Oh, your instructions must have been very clear.
or they're all very smart, or a combination of all the above. Who knows, Elena?
Good audience.
Exactly.
Okay, I'll close the rooms in about 30 seconds and then it'll give them 60 seconds to come back.
Okay. Before you do that, I'll be typing the findings in my PowerPoint. So I just want to make sure that you can see my screen again, right?
Yes.
Yeah, it's not as big, but I guess it's big enough to for people to see, right?
Yeah, I think it's okay.
Yeah, okay, great.
Oh yeah, that's good. Okay, I'm going to close the rooms and they'll have 1 minute to come back.
Okay, thank you.
You're welcome. Okay, it looks like we have everyone back. So over to you, Jeff.
Thank you, Elena. Is Ed back as well?
So yes, great. So what we wanted to do is to just have someone report from each group, and Jeff is going to madly type in the little boxes there. So maybe we could start with group one, and group one, maybe if somebody could tell us about which outcomes would you want to assess?
Well, we talked about some of the outcomes that were already on the diagram that you showed, but we talked a bit about household consumption and food expenditure would be an interesting one to look at, in addition to the primary outcome being HIV. And we also were thinking that the bidirectional association between socioeconomic status and HIV would be a very important one to follow throughout the intervention, just because it would be very difficult to capture the different directions of the relationship. And we talked about a few other things less related to the outcomes specifically, but I'll let others add on to that if they would like.
Okay, great.
Yeah, we were discussing a little bit about the complexity of whether we want the primary outcome to be actually HIV morbidity, or whether it would be the second to last column, because one of our participants highlighted that that's got quite a time lag, so it might be quite hard to measure within the period of a trial. So maybe we focus more on adherence to ART treatment, nutrition, mental health, and empowerment. But we were discussing whether or not they would be primary outcomes or mediators. We weren't too sure.
Okay, great. So any thoughts about the difficulty of doing this?
I'd say it was fairly difficult with just the diagram for prompt, because it did highlight just how complex the intervention was, or is, and I think it brought more questions. So yeah, I think it's a difficult task.
Okay, great. All right, let's go to, thank you very much, group one. Group two?
So we said the mediating outcomes as well as the health outcomes we want to assess. And then we also wanted to assess demographics and loss to follow-up. That was not part of the theory of change. For the primary outcomes, we said morbidity and mortality, HIV morbidity and mortality. Since we were kind of doing our ideal, like we didn't worry about like the sample size for mortality or anything like that. So we just kind of mentioned both since we're not worried about any of those factors. So the exercise, I can't speak for everybody. I thought it was fairly straightforward. The only, I think the complexity that was raised by a couple of the participants was that, what if you have sort of, there's like the mechanism through which primary outcomes are actually impacted. Is it like understanding which pathway through which the effects are coming through? So that was something. I think we talked quite a bit about that, like how and then also how would you assess change in like impact and mediating outcomes and attribute that to the ultimate impact on primary outcomes.
Okay, great. Let's go to group 4, since we know at least one member of group 4.
Thanks for volunteering me, Joe.
Sorry.
Okay. Okay, yeah, so I think the other two group has covered quite a lot. So we also go through like, there's a lot of different outcomes we can potentially measure from the mediators to the final targeted outcome. But I think there's a few questions that we raised. One is that, what is the best way to differentiate between the primary versus secondary outcome? How we should think about that. And also like, given the 2 years timeline, which outcome is feasible, which outcome we can expect to see the changes, and also like within 2 years, which pathway can potentially happen, because some components of the intervention, such as like microcredit or educate or health education, that can lead to changes fairly quickly. I mean, at least that's what I think. But something like agriculture's intervention, that might take longer. And to me, this is a fairly difficult exercise. I don't know what these other think.
Yeah, the time lag thing I found really interesting, because it got me thinking about what about the lag of the effects of things that might have come before the program that might just be lagging into the program when you start the intervention, when you start it. How can you discount, or is there a way of controlling for those types of things, if that makes sense?
That's why we are in the RCT class.
Yeah.
And I think it's the same. It's kind of like, actually, we were looking at potentially things like visits to the health center, but actually that might be, you might actually get more, if you had more visits, is that because they've got microcredit, which means that they've been able to access better healthcare? Or is it that they go less frequently because they're healthier? But we also looked at the nutrition outcomes and things as well. If they bought livestock, did that improve? Like with the microcredit, did that improve their dietary diversity?
Did you decide on a primary outcome?
Not really.
I think we were talking about the sort of the middle ones on your, like in between the mediating factors and the health outcomes, weren't we, at some point? I don't know if we landed on one.
Okay, good.
Okay, great. Jeff just added a note about group 2 that about wanting to know about how many outcomes can be selected. So let's go to group 3.
I can speak for group 3. I think we had a great discussion. Many of the things that we were talking about were these mediating factors that have already been discussed, but some added things that we were discussing was what measure of adequate nutrition that we want to use. So looking at dietary quality and diversity, and also maybe some things that are specific to people living with HIV, such as getting sufficient calories and proteins, changes in body composition if people are on medications. So that was one set of discussions. Another thing was talking about measuring process data. So looking at for our participants, who is purchasing the pumps, who has an understanding of how to use them, how much they're being used. We thought that process data would be useful to measure for this theory of change, as well as potentially measuring some contextual factors to kind of give context to the study. As far as our primary outcomes were pretty standard, just the HIV mortality and morbidity. We might have run out of time at the end to fully discuss that, but those were the two that came up. And then for difficulty, we were just thinking having a little bit more, this exercise might have been a little bit easier if we had some more background on how the irrigation intervention is working and then how HIV and nutrition are interacting with each other. But overall, it was a great discussion.
Okay, great. Thank you very much. All right, fantastic. So a number of issues have been raised, and as we go along, we'll come back to this, and you can see that there's quite a number of outcomes that have been nominated. Some focus on the primary ones being the end outcomes, but some other ideas about some of the intermediate outcomes also, and a couple of votes that this was fairly difficult, yeah, having more information might make it a little easier. For example, knowing that the length of follow-up was 24 months would be helpful in thinking about which things could change over that period of time. And we'll also come back later to the notion that Joe raised about, well, these interventions are not occurring in a vacuum. There's other things that have been going on and could be going on, and how does that affect what we're able to look at. So we'll come back to that and talk about an example of that later. So we'll be using this example throughout the rest of the session, and come back to you with how the investigators went about this. So Jeff, I'll turn it back to you.
Thank you. We're just, so you know, a few slides away from the break, so hang in there. And because we're close to the break, we can just throw some theory at you, so you have a reason to break. So when we started doing this work, we tried to go back to the basic principles, and basically there are two issues that we should be concerned about or pay close attention to when we try to infer conclusions from RCTs. And this is what we'll try to explain on the next couple of slides. So there are, when you conduct an intervention or implement an intervention, there are two possibilities. And this is at the top here of the slide. It is one possibility or option is that if you're interested in dietary intake, for instance, that the intervention did have a positive desired effect and it improved dietary intake. Or it's possible that the intervention was not effective and did not improve this outcome that you're interested in. So this is the reality, what actually happens. And then, of course, what we do in impact evaluation is measurements. We measure these diets, and the measurement does not always correspond to the reality. So here again, we have two possibilities. The evaluation can conclude that dietary intake improved, or it could conclude that it not improve, right? So when you combine these two, the column and the rows, you can see that if the intervention truly improved dietary intake, and we conclude based on our evaluation that it improved dietary intake, we've done a good job. This is perfect. If on the other hand, the intervention did not improve dietary intake and we conclude that it did not improve dietary intake, again, we've drawn the right conclusion. But of course, the two cells that keep us up at night are these two cells. And this is what we need to pay close attention to. And here we get into a bit more of the theory. So it is if the intervention did not improve dietary intake, but because of some issue in our evaluation or random chance measurement issues, we conclude that it actually did improve dietary intake. We call that a false positive finding. So we find a positive result because there is an impact, we say, but it's false because it's incorrect. And this is referred to as a type 1 error. And then the opposite problem is that the intervention actually improved the outcome we're interested in, but we failed to detect it. And this is the lower left cell. That's a false negative. So it's negative. We conclude that there is no effect of the intervention, but that's a false result because in reality, there was an effect of the intervention. We'll talk a little bit more about these two possibilities, and I'll start with the false positive result. So false positive means that we report that there is an effect when in reality that effect was not there. And we'll talk about these false results from two perspectives. One perspective is for one particular outcome. That's the first bullet in bold here. And then the second issue we want to talk about is the evidence base. So if you just take one particular outcome like dietary diversity, we use the p-value of our statistical test to give us a probability of a false positive result. So that's what a p-value in statistics actually tells you. For instance, if we use a p-value of 0.1, that means that there is a 1 out of 10 or 10% chance that we identify an intervention effect when in reality there was no effect. Okay. It is important to also take a step backwards and think about the actual evidence base. When we conduct evaluations, we obviously get more excited when we find an impact than when we find no impact at all. And not finding an impact, which is referred to as a null result, is much less likely to be reported in the literature because it's harder to get published. People are less excited about it, so they give up on writing up the results. But the issue is when results are reported selectively, our collective understanding of what works is biased. Let's just take a bit of a theoretical example. Let's say that intervention A is tested in 40 identical trials, okay? And in one of these 40 trials, we find an effect. So that means that you find an effect in 2.5%, 1 out of 40 of the trials. And if you look at all of that evidence, 1 out of 40, you will say that's likely a false positive result. And your conclusion will be intervention A probably does not work. But if only this one trial is published in the literature and you do not know about the 39 trials that didn't show an effect and are not published, then your conclusion might be that this intervention seems to work because you have identified one trial that showed evidence that it works, okay? So that's the issue with false positive results. And it's obviously clear that we want to minimize false positive results because they do not give us the reality or the truth. And then the other problem is the false and negative results. And just to remind you, false negative means that we report that there is no effect of the intervention, but we actually missed the real effect. We didn't find it, okay? There was one, but we failed to observe it. And when we talk about false negative results, we actually talk about power. And statistical power can be a bit of a confusing concept, but it's actually not that complicated to understand. And I like to use the example in the top right corner of the slide. When you want to study a mosquito, a magnifying glass will probably be very helpful, right? If you want to study bacteria, you need a microscope. And if you want to study a virus, you need an electron microscope, right? So if you want to detect smaller things, you need more expensive equipment. You need more power. And statistical power can really be compared to what type of equipment do you use? And if we try to detect small effects, we need larger sample sizes, more power. We need the electron microscope, and we don't need, or we cannot use the magnifying glass. So statistical power can also be expressed as a percentage. When we have a statistical power of 80%, that means that in 20%, there is a 20% or 1 in 5 chance that a true effect is actually missed. The problem with underpowered studies is that we don't really know what a null effect means. you can compare that with using a magnifying glass to look for bacteria. If you use a magnifying glass and you look at your desk and you don't see any bacteria, there are two possibilities. One is there are truly no bacteria on your desk, or the tool you're using, the magnifying glass, failed to detect them. And we obviously know in this case that you're using the wrong tool. So if you're underpowering a study, it is like using a magnifying glass when you actually need a microscope. So in this particular case, it could be when you find no effect in an underpowered study that there's truly no effect of the intervention, or it could be that the effect is too small for you to detect it with your study. So this is for one particular outcome. Now, again, stepping back and looking at the evidence base, we, as I've explained already, when you use an underpowered study or the wrong tool, you can only detect very large effects. There might be a bacteria that is so large that you could detect it with a magnifying glass, but that bacteria would not be representative of all bacteria we can study, right? So even with underpowered studies, from time to time, you will detect an effect, but by design, that effect will be very large. And it means that if in the literature, when those studies are reported, that again, we will get a very biased view of what the effect of this intervention will be. The effects of these underpowered studies will be large. And when we read the literature and start summarizing what we see, we would conclude that the effect of a specific intervention is very large, where in reality, it would be much smaller. Now, these are the two core problems that we need to solve. Now we're shifting to what the guidance is that is often applied or used by scientists who conduct RCTs of complex interventions. And one of the things that we've discussed a little bit already is that we typically identify two types of outcomes. We have primary outcomes and secondary outcomes. And what is commonly believed is that only the primary outcomes allow the researcher to draw confirmatory causal conclusions. And that trial should only have one or at most a few primary outcomes. And then when it comes to secondary outcomes, very often people think that conclusions for secondary outcomes can only be exploratory, so you can never make strong causal statements about that. And then the guidance is basically not very clear, so there is no clear limits on the number of secondary outcomes you can have. But there are many problems here. When you read the guidance on primary and secondary outcomes, which we did to prepare for this manuscript, it becomes very clear that there is no clear, consistent, or meaningful definition of primary and secondary outcomes. Secondly, there is really no clear reason in any of these documents why causal inference would be different between primary and secondary outcomes. And then related to this, and we'll come back to this after the break, there's a strong belief that if you have more than one outcome, you need to correct for multiplicity. But obviously, it's costly because it makes it harder to detect effects. And since it's costly statistically to correct for multiplicity, people push to limit the number of outcomes. This is something that Ed in that blue table already mentioned, that since we want to understand the pathways, how intermediate outcomes lead to final outcomes, limiting the number of outcomes is not what we want to do because it limits what we want to learn. And this is the last slide before the break. I just want to use a small thought experiment to demonstrate how the current recommendations are inadequate. And I hope this will be clear fairly quickly. Let's assume that we have one trial of a complex intervention and we have identified five outcomes, and we'll call them A, B, C, D, and E. We've agreed on one analytic strategy to analyze the data, and we have two blinded researchers. So that would be Ed on the left and Jeff on the right. I think the next time I need to look for a better picture, but I'll do that for the next learning lab. So Ed and Jeff do not communicate. So Ed starts the work and he decides that in his field, with his background, the primary outcome is outcome A. And the other outcomes are B, C, D, and E. In the analysis, he finds that the intervention positively has a positive impact on all of the outcomes, right? So if Ed was to use the current guidance as people use it, he would conclude that the intervention had an impact on outcome A. That's the strong confirmatory causal inference we talked about. And he would also say that for the secondary outcomes, B, C, D, and E, there is a possibility that the trial had a positive impact on those outcomes, but that would need to be confirmed in a new trial. So that's using current guidance for trials of complex interventions. Now, I do not communicate with Ed. Based on my background, I think that the primary outcome should be B. The others are secondary outcomes. I use the same analysis because we agreed on that. So I also find that the intervention positively affected all of the outcomes. But my interpretation would be that the intervention had an impact on outcome B. And for the other outcomes, A, C, D, and E, I would say there is a suggestive evidence that the trial had an impact, or the intervention had an impact on these outcomes. It's obviously clear that both boxes at the bottom of the screen cannot be true at the same time. We use the same trial, we use the same data, we use the same analytic strategy, and we find very different conclusions. So this is just to demonstrate that there is really no logic in the guidance that is often used for these types of trials. So we will present after the break how we think we should think about primary and secondary outcomes and how we should deal with multiplicity. All right, so this is break time. We would like to ask you to be back in 5 minutes because we've now identified the issues with you, and you obviously do not want to miss out on the solutions and our suggestions. So we'll be back in 5 minutes. All right, let's continue. I hope you had a good break. So what have we done so far? We've tried to explain how inference from RCTs of complex interventions is threatened by false positive and false negative findings. And hopefully we've also convinced you that current guidance does not adequately protect against these two types of false findings, obviously always in the context of using RCTs to evaluate complex interventions. So to build this up or to provide better guidance, we thought trials of complex interventions should really adhere to 3 basic principles. And I'm presenting these principles here on this slide, and then we'll explain each of them in more detail on the subsequent slides. So the first principle is that outcomes should be selected based on the intent of the intervention and on the program theory. And program theory, this is what we looked at for the trial that Ed was describing in Kenya. The second principle is that you should adequately power your study for all of the outcomes that you select that are high priority. And we'll talk more about high priority in a bit. And the third principle is that you need to be explicit and fully transparent about all the hypotheses you plan to test and all of the outcomes you plan to measure. So we'll talk about each of these principles in a bit more detail. So the first principle is that outcomes should be selected based on the intent of the intervention and the program theory. So what really needs to happen is that you need to identify, as we did in the breakout session, all outcomes that the intervention is designed or expected to affect using the program impact pathway. And it's important that here we should not just use or focus on the endpoints. It's also important to understand the intermediate outcomes. It is possible that in the pathways, the program impact pathways, that not all of the arrows, if we can say, flow as well as others. So if we see no impact on endpoints, it is important to understand what happens with the intermediate outcomes. Another issue that is important is that we shouldn't limit to the selection of outcomes to positive or desirable outcomes. We should also keep our eyes open and include potentially negative outcomes. And one outcome, to give you an example, that I've studied in trials is maternal overweight and obesity. This was not something the intervention was obviously trying to increase, but it happened to be an undesirable effect of the intervention we were studying. And it was good that we observed it, because now this is a lesson learned for subsequent interventions. Important here is that you should only include relevant outcomes that will actually be analyzed and reported. And relevant means that the intervention is likely to affect the outcome. And also that evidence on that impact of the outcome will improve our understanding. So it's important to keep that in mind. We don't want to just measure anything under the sky, because it could potentially be part of the program impact pathway. We want to study this carefully and be very smart in selecting the outcomes. The issue is, if you are not careful, is that irrelevant outcomes increases the burden we put on the respondents included in the study, and it also obviously increases the cost of the trial, which are two things we do not want to do. Ed, over to you.
Okay, so this is the same figure that we saw before that we're going to use to organize this discussion. And I wanted to emphasize one of the points that Jeff just made about the importance of identifying all outcomes, because the outcomes that are important to one person or a high priority for one person might be different than for another person. So for example, here, if you're a clinician interested in HIV, you're likely to gravitate towards a lens which puts the focus on the HIV morbidity, which is way over on the right. But if your interest is in agriculture or agricultural livelihoods, or in food security, or in improving empowerment, then your lens is going to be focused in a different place within this diagram, within this impact path diagram. And so identifying all the outcomes is necessary. So next, move forward. So in the trial, what we did was we identified intermediate and end outcomes. So for food security and higher household income and wealth, we identified a number of outcomes. The household food security access scale, so a measure of food insecurity, was assessed, and that could be done continuously and also in categories. Going to the bottom there, household wealth, we created a wealth index, which could also be put into categories, but also meticulously measured total expenditures. And then in the middle, food expenditures could be seen as part of food security or wealth. Looked at total food expenditures, but also food-specific expenditures. And the distinction here between green and blue is, think of the green ones as the ones that we thought were higher priority. Okay, so and one of the things that we tried to do was, for example, for food security, to designate one of the ways of using the outcomes as being the priority and one of lesser priority, so green versus blue. Okay, so next. So then for in the middle there, we have intraretroviral therapy adherence, which was measured, which had multiple outcomes for that. One was self-reported visual analog scale, which we decided was the priority, but then there were others. There was self-report of missed doses and number of doses taken and missed doses. And then we also used the MEMS system to record bottle opening events. And then for mental health, whoops, I don't know why total food expenditures there. Sorry about that. We use the MOS HIV scale and the Hopkins system checklist scale, which is depressive symptoms, as priorities, and also an internalized sigma scale. Then also had a couple of lesser priority ways of using information. Okay, and then next, for nutrition, we had sort of two priority measure outcomes. One is frequency of food intake, and one body mass index, with a measure of body fatness or thinness. And then three lesser priority food-specific frequencies of intake, categories of body mass index, and also mid-upper arm circumference. And then for empowerment, Empowerment is actually a broad construct. So within that, there were really some separate outcomes. One was about self-confidence, one was about sexual relationship power, about gender role conflict, and then another one about intimate partner violence. So all of those were intermediate outcomes for empowerment that were of high priority. And then lastly, for morbidity, the end outcomes was viral load and whether that was suppressed, and then some lesser priority outcomes about hospitalizations, infections, and symptoms. Okay, so taken together, that gives you a picture of the breadth of the outcomes that were assessed in the study. The study was actually more complicated than this because there was a pediatric component that followed children for 2 years after young children, and there was also an adolescent component, but we won't talk about those. So lots of outcomes, and what we tried to do is to think through all of them and then think through which ones were the high priorities. So back to you, Jeff.
Thank you, Ed. All right, so now we've used the program theory to identify outcomes. Now the next principle, as I told you a few minutes ago, is that we need to adequately power for the selected outcomes that are high priority. And when we think about power, remember the magnifying glass, the microscope, the electron microscope. We need to do that to detect an impact of magnitude that is both meaningful and feasible. And meaningful means that the effect of the size we hope to detect is known to be associated with a functional benefit or a harm. And feasible means that it's in line with what the effect size of the intervention could be. And to give you an example to make this clear, when we look at birth weight, for instance, if your intervention, if you could demonstrate a 500-gram effect on birth weight, and just for people who are not familiar with this, babies are typically around 3,000 grams. That's what my children were when they were born. So 500 grams is a massive increase in that. That means that obviously this would be meaningful, but it's not feasible because we do not know of any interventions that would have an effect this large on birth weight. We know that smaller effects are feasible. Something like 70 grams is a meaningful and feasible. So this is something an intervention we know of could have as an effect. And it's also meaningful from a health, a survival, a morbidity perspective. So this needs to be done for all of the outcomes you identify, decide what's meaningful and what's feasible. Obviously, when you try to detect small things, as I explained, the cost will go up. You need a larger sample size. So there comes a moment where you say, well, we would like to detect an impact on this outcome, but we don't have the means to adequately power for this outcome. If that is the case, then you should, typically the general recommendation is to drop those outcomes. Including underpowered outcomes is very common in the field of these types of RCTs, but it's really analogous to hoping to detect a virus using a light microscope. You will not see a virus. So it's not a good idea to include underpowered outcomes in your study. There is one exception that we can think of, and that is mortality. Many studies in the nutrition field include mortality, as these studies are often pooled to conduct meta-analyses, and then the power obviously increases and you can start to make meaningful inferences again. But that is typically not the case for many other outcomes. And then the third principle is that we need to be explicit and fully transparent about all the hypotheses and all of the outcomes. And this is where, there was a question in the chat already about statistical analyses plans. This is where they come in. So trials should be registered before they're started. And trial registration means that you list all of the outcomes you plan to assess. And you should also register your statistical analysis plan before the statistical analyses are actually conducted. Once you have done that, the data come in. You should also report on all of the outcomes that you committed to measuring and reporting on. And that should be irrespective of whether an effect was found or not. You should also be transparent about any underpowered outcomes that you included anyway. It is possible, and that happens, that you include additional outcomes or that outcomes are added somewhere in the process and that are undeclared or were not registered. It's fine to do that, but it needs to be crystal clear in your reporting, in your paper, that these are additional outcomes that were not included when you registered the trial. And any additional comparisons you conduct that were not planned, again, should be reported. What we're concerned about really in here with principle 3 is that what you report needs to reflect the reality. And if you look at this bowl with fish here, you could think of the green fish as outcomes where you find an impact of the intervention, and the red fish outcomes where no effect is found. If you have this information and you decided to report on the 3 green fish and just on 1 red fish, and this is your paper, this is obviously not a good reflection of the reality. The paper here, where 1 out of 4 of these outcomes more or less is you saw an impact, is much more reflective of the reality. So we want to avoid this, and we want to go to this, because this is how we advance the field and how others can learn from your work and not make mistakes in the future by implementing programs that ultimately cannot have an impact on the outcomes we're interested in. Okay, so now over to our definition of primary and secondary and what that means for causal inference. So I've highlighted already the importance of registering outcomes. So you have outcomes that are registered, and in an ideal world, all of the outcomes that we deal with should be registered. But we can also have outcomes that are not registered, maybe things you thought of after registration or halfway through the trial, things you started measuring them. So that would be the second category. And for all outcomes, we've talked about power. So some outcomes are adequately powered, and for other outcomes, you have inadequate power. So combining these 2 dimensions, you have 4 possible types of outcomes, and that will determine what we can say about that. But first, the definition. We think and we strongly believe that primary outcomes should be the outcomes that are declared. So outcomes that you register, where you say before the trial has started, this is what we will assess. And all of these outcomes are also adequately powered. So the effects we could find with this power are both meaningful and feasible. So those are the primary outcomes. And all other outcomes, in our opinion, are secondary outcomes. So there are the inadequately powered outcomes, and there are the outcomes that are not declared, whether they're powered or unpowered or underpowered. All right, now there are 2 possibilities when you conduct the trial. You could find an effect or you could find no effect. So for primary outcomes, when you find an effect, we can draw confirmatory conclusions. And we say the intervention X had an impact on outcome Y. And if no effect is found, you can conclude that the intervention did not have an impact on outcome Y, at least for the size that the trial was powered for. When we look at the other outcomes, the secondary outcomes, you'll see that all of the inference here is exploratory. So for secondary outcomes, you can never make any strong conclusions. So for an inadequately powered outcome, you could say the intervention had an impact on this outcome if you find one, but you'll have to immediately also tell the reader that the size of the estimated effect is likely to be much larger than the average population effect, because you are underpowered, you still found an effect, so it must be an exceptionally large effect. If you find no impact for an underpowered outcome, there's simply nothing you can say. And then for undeclared and not registered outcomes, the table reflects what we have in the top 2 lines, but all of it is exploratory. So if you didn't register an outcome, but it was adequately powered, you find an effect, your conclusion should be that the intervention may have an impact on outcome Y. If you don't find an effect, you say it may not have an impact on outcome Y. And if you have inadequate power for an unregistered outcome, it's similar, it may have an impact, but the impact you document is probably much larger than the average outcome of this intervention. Okay, I know this is a bit much, but at the end of the presentation, we'll share the link to the paper, which is open access, so you can read the paper, and this exact table is in the paper too. All right, so applying primary and secondary outcomes to the trial, handing it back over to Ed to talk you through this slide again.
Okay, so then just take us back to just one example here. You can see that in the bottom right, that the green actually corresponds to what we consider to be primary and the blue to secondary. So for example, for food security, the study was powered. One of the key variables, key outcomes for powering the study was the food insecurity scale in a continuous form, but we will also look at categories, so those are secondary. Same for food expenditures versus food-specific expenditures, which were secondary. And then again, for the wealth index, that there was adequate power for the continuous version of that, but not necessarily for the categories. And so what Jeff is basically saying is that primary outcomes are the ones that are high priority, that are feasible, and for which we know that there's adequate power for those. Okay, so next. All right, so now we've brought ourselves to a situation where we've acknowledged the importance of having many outcomes when we do a complex intervention. And so that raises the question about, okay, what are the consequences of carrying out many comparisons because there are so many outcomes? So when we thought about this issue of multiplicity, which is what it's called, we actually thought about 6 circumstances in which many comparisons comes about. One of them is multiple outcomes, so we're going to put our focus there, but there are some others. So one is about subgroup analysis, when you look at different subgroups to try to see if the effect is greater or not in one group versus another, so that results in more comparisons. When there's more than 2 study arms, I'll come back to that in a minute. Another is when we make initial comparisons, but then that prompts us to then examine some other comparisons based on what we found. Another is when we have multiple times. For example, in the Kenya study, we had measurements at 5 time points. And so if we were to assess the same outcomes at all those time points separately, that would be a lot of comparisons. And then sometimes we want to do interim analyses. Maybe not so much in these kinds of trials, but it could happen that we want to consider partway through whether or not we should continue the study or not, or whether we already have enough evidence. So that would generate more comparisons. So Ramya put a question in the chat box about having more than 2 study arms and the tendency to want to include multiple arms, multiple versions of an intervention, for example, or perhaps to look at synergy among different components of interventions. So to take an example, the MiniMat study, which was a large individually randomized study in Bangladesh about 20 years ago, there were 6 main combinations of interventions for the pregnancy portion of the study. Women could get food either in the first trimester or the second trimester, food supplement, or could get one of 3 formulations of micronutrients. And the hypothesis of the study was that the combination of getting food early and getting the formulation with multiple micronutrients would produce the best outcomes. So that's the synergy between early food and multiple micronutrients in addition to iron and folate. So when that study was planned and the sample size and power were planned, it was planned with that in mind. And that was Ramya's point, that when you have more study arms or want to look at synergy among different components, that's going to require more power. And that fits in with the idea of, as we'll come to, about the best way for handling multiplicity, which is to plan for it and make sure that there's adequate power. So you can go to the next. So we're going to focus here on multiple outcomes for the reasons that we originally talked about. And we're going to try another metaphor, if you like. So imagine we're interested in prison safety and we have a prison with 5 doors. And the probability that a specific prison door fails is about 1 in 100, so 0.01. And what we want to know, though, is not what's the probability of any one door failing. We want to know what's the probability of a breakout, which means that at least one door fails. And so the way we would calculate that, it's not going to be 0.01, it's going to be greater than that. And we can calculate that by what we see at the bottom. The probability that any one of the 5 doors fails is 1 minus the probability that none of them fail, which, if the prison doors are operated completely independently, would be then the calculation shown on the bottom right, which leads to an estimate of about 5% or 0.05. So obviously 0.05 is 5 times bigger than 0.01. So the challenge here is that the probability that any one door fails is higher than the probability of a particular door failing. Now, this calculation was based on the assumption that the doors failed independently. If the doors instead are all controlled by one mechanism, and it's only possible that they'll all fail versus none fail, then the probability would be 0.01. So the real probability is probably somewhere in between. Okay, next slide. So if we bring this back to our situation, as we said before, type 1 error is when we reject a true null hypothesis, an error we don't want to make. And an example would be that intervention A does not reduce anemia, which is the truth, but we conclude that it does. That's a type 1 error. A type 2 error is the opposite, the non-rejection of a false null hypothesis. So for example, intervention B does reduce anemia in truth, but we fail to detect that. So when planning a study, we want to make sure that we have a big enough sample to avoid both errors, and power focuses on the type 2 error. So it focuses on the idea that if there's a true effect, we surely don't want to miss it, because then nobody will be happy. The last thing we want to do, if we're responsible for planning the sample size, is say to our colleagues, yeah, it looks like there was a true effect, but you know what? We didn't have enough power, so the sample size wasn't big enough. Too bad we can't really conclude anything. That's an unhappy situation. So the idea of power is to make sure that the type 2 errors will be small, or in other words, that we have a big enough sample size to detect true effects. Next. Okay, so let's assume that we test a single null hypothesis at, say, 5%. So alpha is 0.05. So the type 1 error rate is 5%. So that means 1 out of 20 times, we're going to wrongly conclude that there was an impact when it was not there. Okay, if we have 2 null hypotheses and do 2 independent tests, this is like the prison doors, each at 0.05, then the probability that we would reject at least one of those turns out to be not 0.05, but it's much higher. It turns out to be about 10%, 0.0975, which is bigger than 0.05. So that means about 1 in 10 times we'll wrongly conclude there was at least one impact when it was not there. So this is the problem that people have been concerned about when they think about multiplicity. Okay, next. So what's shown in the last slide is well known, and it's also well known that when we have a set of hypotheses that are tested simultaneously, that the overall type 1 error rate goes up, that the probability of rejecting at least one of the null hypotheses, even if they're all true, there's no effects, increases. The problem is, and what's often forgotten, is that this simultaneous hypothesis testing assumes a universal null hypothesis, that our interest is in comparing that the intervention has no effects whatsoever across all the outcomes, versus an alternative where there's an impact on at least one of those outcomes. Okay, next. So in this example, that would be like saying, well, we don't actually care about this diagram other than that there's lots of outcomes here. And our interest is in whether there's no effects whatsoever versus at least one effect. That's not a hypothesis of interest. And so that way of thinking about simultaneous testing and therefore dealing with multiplicity in that way doesn't make sense. Next. So what does make sense is to think of it this way. Okay, so we have different outcomes here, food security, and the null might be that the intervention did not increase food security. Alternative, which is our interest, would be that it did increase food security. And for frequency of food consumption, the null would be that it didn't increase food consumption, but the alternative that we're interested in is that it did increase it. And then the same for viral load suppression, that the null hypothesis would be, well, there was no effect on that, but the alternative is that it increased it. And so in this situation, saying that we have these three hypotheses and that we're only interested in whether or not at least one of those holds true doesn't fit at all with the way we think about the way in which the theory of change unfolds in our diagram and the mechanisms that we have in place. And also, if we were to just combine these into one calculation and just look for at least one effect, then we would be ignoring other information that we might have about the study. Okay, so next. Okay, so thinking about correcting for multiplicity is necessary if a set of hypotheses are tested simultaneously. But in the case of this study, the correction would be required if we tested this hypothesis, that it didn't have a positive effect on anything versus that it had an effect on one thing or another thing or another thing. And this joint hypothesis really only makes sense if we define the intervention effectiveness as at least one of these having changed, and that's not what we typically do for the reasons we just talked about. All right, next. So in the case of the study, we've submitted the first manuscript from the study. It's being reviewed, and one of the conclusions there was that this multi-sectoral agriculture and livelihood intervention reduced food security and depressive symptoms and improved self-confidence and social support. And that fits with the theory of change, which was that reducing food security and improving social support would then lead down the road to lower depressive symptoms and better self-confidence, as well as some other outcomes. The study found no effects on the intervention on viral load suppression, but that's because of secular changes in the study area. And Joe Yates in the breakout room raised this possibility. And this is an exact parallel to the issue that Joe raised, was that in the study area over a period of time, the health system has improved its delivery of services for people with HIV. And so what happened during the 2 years of the study is that even in the control arm, that the rate of viral load suppression improved dramatically, so that nearly everybody had a suppressed viral load regardless of what the intervention did. So that's an example where there were lagged effects of some processes that had started before the study even began. All right, next. So then in the literature, it's the literature about correcting for multiplicity in analysis is quite bipolar. There are many articles written where people zealously claim that you must do this. And then there's a number of articles, many articles, including in the Lancet and other top journals, where people are arguing that it makes no sense to do that. It really is nonsense. And so what we've argued is that correcting multiplicity doesn't make sense in analysis because it's costly, because it decreases the type 1 errors, which is the objective, but it's at the cost of increasing the type 2 errors. So in other words, it's throwing away information that is needed to maintain the power to avoid type 2 errors. And so we think and have argued strongly that the solution is not to correct for multiplicity, but to make sure that there's adequate power for all of the primary outcomes. And that means increasing the sample size to do that. But this solution has a financial cost, and it has a societal cost because more study participants are needed. It uses more resources than may be needed, and enrolling more study subjects than needed to answer a research question is unethical. And having an excessive number of outcomes makes transparency and interpretability of findings more difficult. So the idea is to balance these forces. So to the next slide. That what we're saying is that correcting for multiplicity in analysis provides an answer to a mostly irrelevant question. And in the context of our work, we can't think of a situation where doing that is necessary or useful. And so we recommend to not do it unless you're forced to by a journal editor or a reviewer. In my career, I've only had corrections for multiplicity once because I've realized decades ago that it didn't make much sense. And only one article corrected for multiplicity is because a reviewer and editor insisted on it. So if we go to the last slide, what we've recommended in the article is that, and this is kind of bringing everything together now, is first of all, it's really important to specify the theory of change, the program theory, or whatever you want to call it. And that often means working with the intervention and program developers to make sure we understand, if we're the evaluators, what the theory of change is, and make sure that that's specified. The second step is to identify the outcomes and the measures that will be used for those. And in identifying the outcomes, we also want to identify which are the ones that are going to pre-primary, which are the ones that we're intending to have an adequate power for those. And then we have to calculate the sample size to make sure we have adequate power for those outcomes that we designate as the ones that we want to make confirmatory analyses and conclusions about. Step 4 is to register the trial, where we provide in a transparent way for people to see before we even start data collection. What are the outcomes, and in which of those are primary, which of those we're saying we're declaring and will have adequate power, and that we produce a statistical analysis plan, which should be added to the registration at least before the data analysis begins, if not before. Step 5 is to assess program implementation, because part of interpreting the results of a trial of complex intervention is that we understand what actually happened out in the field. And so we want to assess that and add that to the quantitative data. Step 6 is to focus on the declared and adequately powered outcomes, in other words, the primary outcomes, and focus our confirmatory conclusions on those, and limit inference for those that are undeclared and or inadequately powered. And then step 7 is to maintain transparency when reporting outcomes, so that we report everything. One of the challenges that we have is, can we put everything all in one manuscript? And that's a topic we could debate in the discussion if you want. But the main point is to be transparent about when reporting outcomes, so that we're being honest about, okay, which ones were declared, which ones were primary, which ones were secondary. And even if we don't find effects where we were hoping to, that we report those. And if we find harms, that we report those as well. So all of this is written about in the article that was just recently published a few weeks ago, which is listed here, and it's been put in the chat, and we encourage you to take a look at that. Okay, so now I'll turn it over to Shelley, and Shelley is going to coordinate the question and answer period.
Thanks very much, Ed, and thanks to Jeff as well. Thanks for the very informative session. We have about a dozen questions in the chat that I'm going to pose to you, and I know you've both taken an attempt at a couple of them to try to answer a couple of them, and we'll make sure that those questioners are happy with the answers and don't have remaining questions. So Ramya asks, and I know some of these have been partially addressed, but I think they're important points to underscore for everybody to understand. How many outcomes can we select for primary and secondary outcomes? And I know you've addressed that, but it might be good just to come back to that. And then she also asked, comments that there's been a recent push towards multiple arms and synergistic effects for efficiency. However, the power to detect synergistic effects requires really larger sample size, maybe 16 times as high. And when we find synergistic effects that are not significant, very few studies talk about lack of power. Instead, it's interpreted as null effects. Is there guidance for multiple arms and complex interventions for both sample size and reporting?
Okay, maybe I'll take a stab at it, and Jeff, you can add. So on the part about how many outcomes, I mean, it really depends upon the situation. I'm working with, for example, the team that's doing the Suhara, that they just started the data collection a few days ago of the Suhara intervention in Nepal, which is a huge program covering a good portion of the country. It's multi-sectoral. There are a lot of intervention components, and therefore there's a lot of outcomes. And it's important that there are outcomes that represent all of the different components of the trial and the different aspects of people's lives that could have been affected by the trial. So that's a situation where we're going to have a lot of outcomes. This Kenya trial we talked about also had a lot of outcomes. I think one useful guideline is think about an outcome as a construct, and what we want to do is to think about what are the important outcomes and then have one measure that's the primary measure to go along with that primary outcome, not to have 7 different measures of depression symptoms, for example, to choose one and stick with that to limit the number of outcomes and comparisons that are going to be made, but while still making sure we cover all of the interests in the trial, because the major issue here is how can we learn as much as possible? Doing trials is a learning exercise. It helps us gather information so we can learn and then do better in the future. I commented before about when we have complex interventions with multiple arms, and if we're interested in combinations of those arms, then absolutely the sample size requirement is going to be much higher. And so there's a trade-off there. If you have 6 arms in a study instead of 2, the sample size requirements are going to be much, much larger. And so some trade-offs might have to be made in that regard.
Thanks, Ed. Jeff, anything that you want to add?
No, Ed covered it quite nicely.
Okay, great. Thank you, Ramya, for those questions. Reshma asked, are overpowered studies equally problematic? And Ed covered that, I think, in that second to last slide when he showed both the financial costs, impact, participant burden. Reshma, do you feel like you have a good grasp of overpowering a study, or yeah? Is there anything else you'd like to add to that?
Yeah, I think I meant more kind of on the effect size, because what Jeff was kind of talking about for underpowering was related to detecting large effects when in reality they might be small. So what happens in the case of overpowering?
Yeah, when you overpower, you don't have anything similar. You will be able to detect effects that may not be meaningful anymore. You're using a very costly microscope to detect something that you could find with a magnifying glass, so to speak. But your funder may not be terribly happy because you're basically using resources that you shouldn't be using, because you could detect what's meaningful with a smaller budget. So there are ethical issues there as well. Yeah,
I think I agree with all of that, but there's another perspective about it, and I want to tie this to a question that Kendra Bird asked about the situation where there's students who use the data that have been collected, and others come in. I mean, there's definitely trade-offs to be made. Sometimes we collect what could be seen as an overpowered sample size because we're interested in secondarily maybe finding certain subgroups that may benefit more than others, or we're imagining using the data to pursue some other questions because data collection is really costly, and if we can use the data from a trial to answer some other questions down the road that maybe don't use the trial design but use the data. So it's important to think about it for the immediate need for the trial, but maybe also for what are some broader uses. But those are difficult trade-offs to work through.
Can I just add to that question, because we are obviously not suggesting that the student add-on projects should be stopped. I think if those student projects focus on outcomes that were not registered but measured anyway, then I think all that is needed is that in the write-up of those results, it's made clear that those are outcomes that were not registered. So we need to be careful about the inferences made based on the analysis. Because as a reader, you do not know if there were 150 outcomes that were measured but not registered, and the student just selected the 6 where an impact was found. You start to bias the evidence base, so to speak. So that's why these are exploratory analyses and they should be reported as such.
Great, thank you very much. The next question is actually what I was addressing for from Ying Lan. Can you explain why correcting for multiplicity is costly? And that's the one where Ed talked about that. And Young, do you have any follow-up questions, or is that clear to you now after Ed's presentation?
Maybe I can add a comment about this. I have struggled to put this into words, but I'm going to try. I think that doing adjustments for multiplicity and analysis is a simplistic solution to a problem where inherently the situation is complex. And we humans can do a much better job of integrating information in a meaningful way about what it means for different outcomes to have shown an effect at different parts of the impact path diagram. We can do that in a sensible way and include information about what we know about the context and the situation that's occurred in a much more sensible way than just turning it into a simple mathematical calculation. And that's long been my objection to doing adjustments for multiplicity.
Great, thanks. And Yun confirms that the answer is clear now. So thanks, Ed, for that. Next, Jeff, you answered this partly. Kendra asked, do people recommend pre-registered statistical analysis plans? And I think the answer was clearly yes. She asked what are the best platforms for doing this. Do either of you have any recommendations on that?
Not on specific ones. I mean, I've always used clinicaltrials.gov, but other institutions, WHO has also a trial registration platform. A few other institutions have those as well. I don't have enough experience to say that this one is better than the other one. To me, the most important thing is to register your trial before it's conducted.
Great, thanks. Kendra,
if I could just add, Shelley, there's another benefit for having a statistical analysis plan, which was definitely the case in large trials that I've worked on, which is that there's many investigators, and it gives the analysis team a way of building consensus among all the investigators before going into the analysis, so that when an investigator says, well, why don't we try this? Why don't we try that? Why don't we try it this way? Why don't we do it this way? You can say, as the people responsible for doing the analysis, well, you know, that's not in our statistical analysis plan. That is a major benefit.
And I'll add just one quick comment to that. It also protects you against reviewers. We've had situations where reviewers suggest, could you run this subgroup analysis? Could you do this? And we say, no, this is not part of what we registered to do. So we're sticking to our plans here.
Thank you. We just have 4 minutes, and I think more questions are popping up here. So we're going to try to go really quickly through these. Ramya, you asked a long-ish question on different disciplinary lenses and how they view different interventions and having different primary outcomes. She's wondering if part of the recommendation on conducting complex RCTs could include that ethical review boards are necessitate trial and statistical plans registration. Easy to make it a requirement rather than just saying best practices. Second, planning requires money, so donors and researchers should account for this required component in the proposal and plan for a year or year and a half for this process before complex RCTs start. Any comments on that suggestion, Ed or Jeff?
I'll comment on the first one, ethical review boards. This may depend upon context, certainly in the US, ethical review boards are responsible for reviewing the human subjects issues, the ethical issues involving human subjects, period. That's their job. It is not their job to deal with any other issue. In some other countries, studies have to be reviewed and approved for their scientific aspects, not the ethical aspects, but the scientific aspects. And it may be that there it makes sense to do it, but no, not part of an ethical review board. That's outside their mandate or their training.
Okay.
I agree.
Aisha Gambo asks, will journals accept manuscripts easily that reported that an outcome was adequately powered?
Will they accept outcomes that are adequately powered?
Manuscripts.
So
that
I mean not adequately powered. I made a mistake. That not adequately powered.
Okay.
I review for the Journal of Nutrition, and it's one of my favorite comments when I review papers that the authors are not saying anything about power. So I think it's an issue that does not receive sufficient attention in journal and article reviews, and that I would hope we would pay more attention to. So you can still get away with this today.
Unless you're the reviewer.
Yeah,
right. But I think it's important to emphasize what Jeff said earlier, which is we're not saying don't include that information in a manuscript. We're saying be transparent about it. Be transparent that this is what we found, but we didn't have adequate power, or we didn't plan adequately for the power for it.
Be transparent. Yeah.
Yep.
And the next question is about that. This is from Nishmeet. It may happen during the course of a program that the intervention changes may be expanded or contracted as we saw during COVID. So are there any suggestions for changing outcomes or adjusting for initial power calculations as the trial proceeds?
Yeah, we don't work in labs. We work in the real world, and all sorts of things don't go the way we plan it to be. I think, again, the key word here is transparency. It would be silly to completely give up and say we can't stick to what we originally planned or registered, so we're giving up entirely. I think it does make good sense to make the best of a situation, but as long as it's clear and you can actually change a trial registration and it's dated when you made the change, as long as you do that and you're very transparent and you write up that the intervention changed, that your way of measuring your outcomes changed, I do not see there is an issue, because then the reader can interpret what you did, how it was analyzed, why changes were made, and how good the evidence will be.
We're at time, but there are 4 or 5 other questions. Elena, what do you suggest?
Perhaps people can reach out to you all with further questions for the future. Is that okay?
Absolutely. We have lots to learn from all of you who are out dealing with these issues, and we'd love to hear from you.
Well, I want to say thank you very much from the ANH Academy to our 3 facilitators in this session. This is our last learning lab of the week, and we hope you'll join us for some of the research conference next week. And to our facilitators, if you're willing to share your contact information in the chat, please go ahead. I bet people would have follow-ups for you.
Thank you, everybody. Thanks, Shelley.
And Jeff as well.
Yeah, thank you.
Thank you. Bye-bye, everybody.
Bye.